The topic of this short video lecture has to do with how our brains filter signals coming in from noise that comes in. So first we want to just remind ourselves that we talked about long-term potentiation and mechanisms for how we make synaptic connections more excitable. So today we're going to talk about how we can have a series of synaptic connections to create a filter. So long-term potentiation happens in the hippocampus, and today we're going to be talking about the hippocampal filter. Now we're going to filter out. The picture here says at the bottom that we can distinguish a single voice in the middle of a noisy street. So I got a little cartoon picture of that. This slide just shows you sort of a deep brain structure called the hippocampus, the place that we're going to be working and looking at with signals coming in, how we filter them to be aware and or to have the potential to make memory. The hippocampus consists of what's called the trisynaptic circuit. It involves cells in the dentate gyrus, the CA3 region, and the CA1 region. And we're not going to be spending time for our topic to be talking about the different pathways that are shown in red, but just to have an understanding of the communication, because we need to be familiar with these parts here. Signals that come in will, if they're really moderate to high signals, seem to be able to pass through this circuit, and signals at relatively low frequencies coming in seem to get filtered out. So we've already discussed the two forms, break down the two forms of the glutamate receptors, the metabotropic G protein coupled and the ionotropic glutamate receptors. And for today we'll be focusing on 5 and 7 as two that we'll be able to see play a role in this phenomenon of the hippocampal filter. Now before we get to the hippocampus and talk about the filter, it is going to be important for us to understand this exocytosis of the neurotransmitter. And so you can clearly see here in the synaptic vesicles, little cartoon pictures of our neurotransmitter here, and they fuse, and as the action potential travels down, we're going to focus on these calcium channels. So it's important for you to understand that these are voltage gated calcium channels, and they appear at the axon terminal. So let's write that on here. They're voltage gated. Now obviously they're going to open in response to voltage, so as the action potential is traveling down, you'd have depolarization in this little piece of the membrane here that I just kind of scribbled on, and that would cause that channel to open up. Erase that. So it's a voltage-gated signal for calcium to come in, and then once inside, all of this fusion and release, these are all, it's just a chemical process through actually calmodulin and a couple other things you already know. But important for you to understand that we're going to really be looking at calcium coming in too. So one interesting phenomenon that, again, we're not quite to the filter, but it's called presynaptic depression. So if we have high amounts of neurotransmitter release, like shown in this picture, okay, so the blue one is going to be the presynaptic neuron that's releasing the neurotransmitter, and the green one down here is the postsynaptic target. And so But you can see, if we were to sort of make a little drawing over here, we'd have these receptors in the presynaptic membrane, and the presynaptic membrane is also releasing the neurotransmitter glutamate. Okay, and across the synapse would be the target and the receptors for that glutamate. So here's my target, and I'll put a little receptor, R for receptor there. And so the glutamate's going to stick and bind to this spot. Now, it's important that you understand that it says here, with high amounts of neurotransmitter, a lot of it, that neurotransmitter is released, and with enough of it, it can come back over here and it can bind to this receptor, and this is a metabotropic glucose receptor, so it's called an autoreceptor because it's in the presynaptic membrane. And the autoreceptor is going to activate a G protein process to come over here and stop the calcium-mediated release of neurotransmitter. That's all emanating from the presynaptic side, and it's depressing the response in the postsynaptic target, so it's called presynaptic depression. So now with this figure, what I'm going to do is try to really just summarize and see if we can understand the different glutamate receptors playing a role in this figure. So first we'd have, from our chart that we saw previously, the metabotropic glutamate receptor 2, and this one's going to be a slow inhibitory. You can see from the GI there. The 3, which is also in that slow inhibitory group on the glial cells. 5, which is slow but excitatory, and 7, which is slow and inhibitory. We see the little minus sign there. Now, if we take a look at the dynamics of what's happening in this picture, first, calcium-dependent exocytosis. You could see it doesn't show the calcium, but you can see the exocytosis taking place here and down over here. Now, if we have an increase in glutamate coming from the presynaptic terminal, we've got the only example here of the ionotropic glutamate receptor in the membrane. Glutamate binds to it, and here we see it's nonspecific for sodium or calcium to rush in. Now, if we look at the important glutamate receptor 7 in this picture, you say if this presynaptic terminal down here was firing AX potentials, the glutamate receptor is in the presynaptic membrane. It's like an autoreceptor. So through a G protein coupled event here with the G protein, it inhibits adenylate cyclase and reduces cyclic AMP, inhibits voltage-gated channels, and gets less voltage-gated calcium coming in, so you get less calcium-dependent exocytosis, and you reduce this activity here, so you get less binding into the synaptic space. This neuron is inhibitory, so it's releasing GABA, and GABA is ionotropic. It binds to a receptor on the postsynaptic membrane, opens a chloride channel, and so chloride goes in. So let's just take a look. If the autoreceptor was activated because of this constant inhibitory signal that was being sent, excess neurotransmitter could shut this pathway off. The glutamate would have to float over here and shut this off. not the GABA. This is for glutamate, this one. So we finally come to describing the filter. And here's the example showing 3 cells within the hippocampus. So you've got your granule cells here and your pyramidal cells here. And so the pyramidal cells are what are looking to receive this information. Now, under normal circumstances, with signals coming into this system, you'd have some signal originating in this direction, coming down here to have a synapse on the pyramidal cell that's going to release glutamate, and the role here would be to excite the pyramidal cell, and the role here would be to excite this interneuron. So I'm going to put a plus there because I can't write excite there. The interneuron, when it's excited, is also going to provide some inhibition to the pyramidal cell, say through GABA. So if the levels here are low, at low levels, you get excitation coming in from up above here. You get inhibition coming here. So the excitation and the inhibition cancel each other out, and in a sense acting like a filter. It's important to understand with low to moderate levels, that's sort of a key feature here, because the glutamate that's being released here is low to moderate and here is low to moderate. Okay? This question says, what does the interneuron do? It shows right here. It inhibits the pyramidal cell. So, in looking at the points typed on the screen here, where would you need the metabotropic glutamate receptor 7 autoreceptors to allow for weak signals from the granular cell to the pyramidal cell to be blocked, and strong signals from the granule cell to get through to the pyramidal cell? So again, we're gonna just describe this filter. We just described it in the previous slide for how at low levels you would have, in a sense, canceling out at the 2 inputs to the pyramidal cell. The input from the granule cell in red is excitatory, and the input to the pyramidal cell from the inhibitory neuron is shown in black, and they cancel each other out. Okay? Now if you have high levels of input coming in to this cell, you would get really strong signals for glutamate, both at the level of the granule cell to the pyramidal cell right here, and at the level of the granule cell to the interneuron shown right there where I circled it. So these high levels here would look something like this. And really what we're talking about is putting the metabotropic glutamate receptor 7 on this presynaptic terminal right here, which says then if the levels were high and there was lots of neurotransmitter coming out, remember at moderate levels the red axon terminal from the granule cell excites the interneuron and leads to inhibition, and it cancels out. At high levels, we eventually get the glutamate receptor 7 coming in here and blocking neurotransmitter release, thereby really reducing, eventually reducing the amount of neurotransmitter here in the synapse. If we reduce that, then we have basically a very quiet inhibitory. So again, with high levels here at that synapse, you get the autoreceptor shutting off the release of neurotransmitter from the granule cell. You'd have no activity here in the inhibitory neuron to the pyramidal cell. So with high levels of stimulus coming into the granule cell, this excitation dominates. We remove the inhibition down here, and the signals go through to the pyramidal cell. So high signals go through, and how relatively low signals get filtered out. So in conclusion, and to have the ability to sort of think of critical thinking questions, let's just really go back and do a quick review. And you could watch this video and look it over as many times as you want to. But I think the first thing to understand is that we're dealing with some the important role of the metabotropic glutamate receptor 7. That this is, when acting as an autoreceptor to glutamate, can eventually lead to a decrease in neurotransmitter release. We saw a couple different examples of that. So it's basically blocking this calcium and working in conjunction with calcium, right, to block the calcium mediated exocytosis. And understanding how this receptor would work, in the last few slides it says at low levels there's basically the granule cell coming through to talk to the pyramidal cell, and that leads to excitation, and the granule cell talking to the inhibitory neuron to that same pyramidal cell. And at low levels, in this color, and they filter each other out. The excitatory part at the top and the inhibitory part at the bottom filter out. If we switch then to summarize here, that with really high levels of signal, we would get lots of excitation from the granule cell directly to the pyramidal cell. We would get lots of excitation here. And this is where the glutamate receptor 7 is going to shut off. Right here, it's going to shut this off. And so we're going to remove the inhibition, in a sense, taking this one out. So at high levels, shown in red, we end up with this tremendous excitation. So we can see the levels of the filter, and that's the important stuff. So make sure you can understand this and get with me if you have any questions.